Hello, grade 12 students. In this video, we're going to continue with our chapter 9 about the nervous system. And we'll move to document 5, synapse, structure and function on page 192 on your book. This document already, we discovered the different types of synapses. We have in neuronal synapses between two neurons, or neuroeffector between a neuron and effector. Maybe it's a muscle or maybe it's a gland. So we talk about the function of the calcium ions whenever they enter into presynaptic. They cause the increase in the level of the acetylcholine, causes the propagation of the nervous message, and we talk about the effect of the acetylcholine. So whenever we have acetylcholine, high level of acetylcholine, this will cause an increase in the level of the potential difference. So we said that the acetylcholine is responsible for the increase in the level of the potential difference. So we said that the potential difference is directly proportional to the amount of the acetylcholine. Also, we talked about the action potential arriving at the level of the presynaptic terminal. At the presynaptic terminal, what will happen? About the presynaptic terminal, we have the action potential arrives. So calcium ions, they are going to be rushed into the presynaptic. This will lead to the exocytosis of the neurotransmitters from the secretory vesicles. This exocytosis will be inside the synaptic cleft, binding to the receptors and causing either IPSP or EPSP. PSP is post-synaptic potential. So IPSP, we're going to talk about this in details in document 6. So IPSP, it's inhibitory post-synaptic potential. So it has an inhibition. So let's see what will happen in IPSP. Or if it's EPSP, excitatory post-synaptic potential, we will see what will happen. So these, we covered most of them. So we said that the nervous message is propagated from the presynaptic to the postsynaptic. If we did an excitation or a stimulus, an effective stimulus on the pre, the message is going to propagate toward the post. And if we did the stimulation on the post, it won't go back to the pre. So this means that the message, it moves in a unidirectional manner. It moves from the presynaptic terminal toward the presynaptic terminal, toward the postsynaptic terminal, sorry. And we said that we have an action potential reaching the presynaptic terminal. It provokes, it facilitates, it causes the entrance of Ca2 plus of calcium ions. This entrance of calcium ions in the presynaptic terminal will cause the message to be transmitted from the pre toward the post. This entrance of Ca2 plus, it causes what? It causes the exocytosis. You have to know this word, exocytosis, the release of the acetylcholine, the neurotransmitter, into the synaptic cleft. This will have a role in the conduction of the nervous message toward the postsynaptic. And we said that the amplitude is directly proportional to the amount of the acetylcholine. So as we increase the amount of the acetylcholine, the level of the potential difference will increase. So this is the conclusion. Action potential, whenever it reaches the presynaptic terminal, it causes the entrance of Ca2 plus. This Ca2 plus, it provokes the exocytosis of the acetylcholine, ACH, is the symbol of acetylcholine, into the cleft. This will lead to the propagation of the nervous message from the presynaptic terminal toward the postsynaptic terminal, as well as we will have an action potential. And you have to know that the potential difference is directly proportional to the level of acetylcholine. Here, the last part that we didn't cover it in the previous session is about the difference between the voltage-gated channels or voltage-dependent channels and chemical-dependent channels. From its name, voltage-dependent channels. They open and they close, opening and closure of these is according to the difference in the potential difference in the membrane potential. It's different in the voltage. That's why they are called voltage-dependent channels. Whereas chemically dependent channels, they open and they close according to what? To the binding of specific chemical substances to the receptors that are found on these channels. And in class K plants, they are voltage dependent channels. Okay? So these are the difference between voltage dependent and chemical dependent. Chemical, they open and they close due to the binding, of chemical substance to receptors found on these channels. Now we move to document 6 about the integrating properties of the nerve centers. It's page 194 on your book. So integrating properties of the nerve center. In the nerve center, a neuron is connected to different types. It receives several afferent. Afferent, it's what? It's the sensory. It's the afferent, it's a sensory neuron, and elaborates only one efferent. So it's connected to a multitude of other neurons. It receives several afferent messages. So they receive several afferent sensory messages, and they elaborate or they emit only one efferent. Here, if you don't know that afferent is the sensory and efferent is the motor, you won't understand the sentence. So these are the notes about the efferent and afferent neuron. Who can tell me what are the different types of neurons according to their function? There are three types. First type, it's called the afferent or the sensory. Second type, Muhammad, is called the efferent or the motor. Third type, can you give me the name of the third type?
There are three types: afferent, and interneuron. The
Interneuron
afferent is
or
sensory.
afferent is the sensory, efferent is the motor, while
Is the motor. The it's the connection between both neurons for the
okay, it's
central nervous
connection. It has another name. Can you give me the other name of this neuron?
system. Yes,
It's called associative neuron.
neuron.
Okay, so we have three types of the neurons. Sensory neuron, which is called the afferent neuron. The second one is called the motor neuron, which is called efferent. And the third type is called the interneuron or the associative neuron. It's found mainly in the brain and the spinal cord, and it's like a link between the sensory and the motor. So it's like a link. Let's now talk about excitatory and inhibitory synapses. Document A, which is this document, this is document A. Document A represents the cell body of a motor neuron in the anterior horn of the spinal cord with synapses, about 50,000, 80 to 90% of which are found on dendrites. So that provide this cell body with information coming from other parts of the nervous system or from the muscular system. So by introducing a microelectrode, ME, this is the microelectrode that is introduced in the motor neuron, the oscilloscope, it records what? A potential difference. It records PD, potential difference of about minus 70 millivolt. What is this minus 70? This is the resting potential. It's equal to minus 70 millivolt. Following an effective stimulation. So we have an effective stimulation. This motor neuron, it responds by action potential that originates in the cone of implantation. This is cone of implantation, this one of the axon. So we have effective stimulations. They are applied separately on the presynaptic fibers, number 1 and number 2. Here we have effective stimulations are applied on the presynaptic fibers number 1 and presynaptic fiber number 2. Effective meaning that they will lead to the appearance of the action potential. Effective stimulation S1 and S2 on the fiber 1, which is here, and effective stimulations, they are applied on the presynaptic fiber number 2. Their corresponding responses are recorded at the level of the postsynaptic neuron. So they are recorded at the level of the post. The stimulation applied on fiber 1, so pay attention, this one, this is the fiber 1. So the one that's applied on fiber 1, this leads to hypopolarization, leads to depolarization. This is one, it's shown in document B. While the similar stimulation that are applied on fiber 2, these one on fiber 2, they lead to hyperpolarization. First of all, let's start step by step in the first one, in fiber 1. Here in fiber 1, we have hypopolarization. We said that polarization is the negativity. Hypopolarization, decrease in the negativity. So we are moving toward the positive. While in the second one, in fiber 2, we have hyperpolarization. So what does this mean? Hyperpolarization. It's decrease It's increase in the negativity and decrease in the positivity. Hyperpolarization is negative more than the normal, which is seen in this figure 2 of document B. So this one leads to hyperpolarization. So it's negative more than the usual. Whereas in document 1 of B, we have hypopolarization. So we have depolarization. So we are moving toward the positive. So here we can say that excitatory postsynaptic potential, it's here. Since you move toward the positive, excitatory postsynaptic potential, EPSP. E excitatory, P post, S synaptic, P potential. Whereas in document 2, we have inhibitory postsynaptic potential. Since it decreases toward the negative, we have hyperpolarization. So this is referred to as IPSP. In the document 5, we talk about PSP. We only talk about the PSP, post-synaptic potential. But in this document, I said that we are going to move deeper. Either it's an excitatory post-synaptic potential or it's an inhibitory post-synaptic potential. So here we move in document 2, and this one, we move toward the negative. So the potential difference or the membrane potential, it decreases more than the normal. It decreases below the resting potential. So here we have hyperpolarization. So this is known as inhibitory, I, P, post, S, synaptic, P, potential. So we have here I, P, S, P, inhibitory, post, synaptic potential. While in the figure 1, we have EPSP, excitatory post-synaptic potential. Since we have hypopolarization, or you can say we have depolarization, and it's an excitatory post-synaptic potential. Moving now to this figure, or talking about the excitatory or the inhibitory characteristics of a synapse, it mainly depends on the receptor that's found in the membrane. For example, acetylcholine, it's an excitatory if it's found in a synapse, and it can be inhibitory in another synapse. It's excitatory when it increases the level of Na plus entrance inside the cell. So whenever Na plus they enter, they open the channels of the Na plus, they enter inside the cell, so the internal will become less negative. So it will move toward the positive. And this is called the hypopolarization or the depolarization. While in another synapse, maybe the acetylcholine will increase the permeability of K plus and Cl minus to enter inside the cell. So whenever they enter, the internal will become more electronegative, and it can be inhibitory. So here, inhibitory or excitatory, it depends on the synapse. So excitatory when it increases the entrance of Na plus. So acetylcholine can be inhibitory in one synapse and excitatory. Excitatory when it allows the entrance of Na plus, sodium ions. So whenever they enter inside the cell, it will become less electronegative, and this will lead to the depolarization or the hypopolarization. However, if it allows, it increases the permeability of K plus and Cl minus, this will lead to the increase in the negativity inside the cell, and this will lead to the hyperpolarization. It will be more electronegative. Moving to paragraph 2, integration of information. Here, I'm going to talk about two different types of summation, and you will know the meaning of summation. We have two different types of summation. We have something called temporal summation, and we have something called the spatial summation. Let's start step by step. Integration of information in paragraph 2. So transmission of the messages is due to either too many fibers that function in parallel with each other, or to repeated impulses of the same nerve fiber. Here we have two mechanisms. They present respectively the temporal or the spatial. Either too many fibers that function in parallel, they function together, this is called the temporal. While repeated impulses, this is called the spatial summation, and we will give the definition of these, spatial and the temporal. If fiber 1 of document B. So fiber 1, we said that it has EPSP, if you remember. Fiber 1 of document B, this is fiber 1 of document B. This one, fiber 1 of document B, it has EPSP, excitatory post-synaptic potential. Sure, we have to work on this since we have to need to have an action potential, excitation. So it triggers an action potential resulting in a temporal summation, EPSP. So temporal summation. What's the meaning of temporal? Let's start the definition of both temporal summation and spatial summation. Temporal is the summation of two nervous messages. So two messages, they come from the same synapse and we add them together. This is called the temporal summation. However, the spatial one, summation of two nervous messages that come from two different summations or for two different synapses. So we have two synapses, they send the message, the same message. Whenever we add this, it came from two different synapses, we call it a spatial summation. However, if the summation came, the messages they came from one synapse and we have more than one message from the same synapse, it's called the temporal summation. So temporal summation here is in figure 1. Figure 1, or document C, temporal summation, they came from one synapse. So two or more messages, they came from the same synapse. So here from the same synapse, two or more messages they come. So we have two of them, they are added. So we add these messages. We have summation of the message, addition of the message. So when fiber 1 and fiber 2, they are stimulated simultaneously, the respective postsynaptic PSP add up so that they can attain the critical hypopolarization. They will reach the threshold needed to trigger an action potential in the postsynaptic. This will result in spatial, since they come from two different synapses, synapse 1 and synapse 2, for example. So they have spatial summation of the postsynaptic potential of each stimulant. This is shown in document D. So here we have S1 and S2. So S1, it has excitatory, S2, it has an inhibitory. So why did the message reach this level and not this one? What is the difference? Who can tell me? What is the difference? Why do we have in figure D, in the spatial summation from S1 and S2, synapse fiber 1, fiber 2? One of them is excitatory postsynaptic potential, and the second one is inhibitory postsynaptic potential. Imagine that you have a car. This car are moving and you use the brakes. استعملت الفرامل, so the speed will decrease. So excitatory post potential and the excitation. So the potential difference is increasing. However, if we have inhibition, so it will decrease the excitation. It will be excited if the excitation is more for sure, but it will decrease since we have something that inhibits the propagation of the message. However, in the first one, in the temporal summation, they came from the same one, from the excitatory postsynaptic potential. So whenever we add them, all of them, they are excitatory. So the nervous message, the potential difference will increase. And this is the temporal summation. Yes, Muhammad, you raise your hand. Do you have a question?
No, it's fine.
Okay, so here, excitatory postsynaptic potential, it's excitation. Inhibitory, it's inhibition. It leads to hyperpolarization. So whenever we add them, sure, we won't get this result here in the case of only excitation, since we have inhibition of the nervous message. So the inhibition will stop the propagation. For example, if I gave you a question in the exam, I will give you a question similar to, let's say, I will give you a question that we have three different synapses. Okay? Three different synapses. Synapse 1, synapse 2, synapse 3, synapse 4. And the nervous message is propagated from synapse 1 to synapse 2 to synapse 3 and to synapse 4. So the nervous message is propagated between the four synapses if we did a stimulation. So these are excitatory synapses since they move. So we have an action potential in the four. Why? If between two synapses, the nervous message or there is no an action potential, this means that these two neurons, they have a synapse in the middle between them. This synapse is going to be an inhibitory one since it inhibits the propagation. So you have two neurons. If you apply one stimulation, if you apply a stimulus on one of the neurons and the second neuron is connected to it due through the synapse. So if the message is propagated from one neuron to another, this means that this synapse, this connection is an excitatory one. It allows the propagation of the nervous message. However, if the message is not propagated from one synapse to another, this means that this is not an excitatory, this is an inhibitory synapse. So pay attention to this note. This is a very important related to the temporal summation and the spatial. So they said in probing the documents in part 1, what are the different types of synapses shown in document B? So these are in document B. So in document B, whenever we have EPSP, so what is the type of the synapse? Yes, Badia, if you have excitatory post-synaptic potential. So what is the type of the synapse? Is it an excitatory or inhibitory?
Excitator.
And while in second one, Al Hariri, if we have IPSP, so the synapse is what? Al Hariri is not here, regard. Yes, Rama, what do you think?
Inhibitory.
Inhibitory.
Inhibitory. While we have IPSP and we have hyperpolarization. Part 2, what is the ionic behavior responsible for EPSP and IPSP? Already we explained this in this paragraph. We explained question number 2 in this paragraph. Whenever we have EPSP, excitatory post-synaptic potential, so Na plus channels will open, so they will enter inside the cell, it will become less electronegative, so we will have excitation. While if the Cl minus or K plus channels, they open, they enter inside the cell and will be more electronegative, so this will lead to the increase in the negativity. Part 3, I need only the part 3, part C. Parts A and B, I'm going to give definition as notes for them. So I'm going to move only to the role. So let's move from the document page 195. So synapse of document B1 is excitatory. So why? Since it shows hyperpolarization. So hyperpolarization of the membrane potential, it shows decrease in the negativity, depolarization. Prerequisite and essential for generation of the action potential. Since we don't have an action potential, so it's preparation to have an action potential. While the synapse of document B2, it's inhibitory one. Why? Because it shows hyperpolarization. So increase in the negativity. This will make the membrane away from the threshold of depolarization, and so there is no action potential recorded. How the difference between EPSP and IPSP according to the channels? So if sodium chemically gated channel is open, so this will allow the inflow the entrance, okay, into the post-synaptic cell. This will make the anterior less electronegative, meaning that more electropositive, and we have EPSP, excitatory post-synaptic potential. While if potassium or chloride channels they open or allow the entrance of the flow, if chloride, this will cause the interior becomes more electronegative, and we will have IPSP, inhibitory post-synaptic potential. Motor neuron, what is the role of the motor? It has integrating role. What does it do? Either it receives different afferents and it will do algebraic summation and will give one efferent message. Okay, so this is the role of the motor neuron. It receives either many and add them and give only one efferent message. Okay, one motor message, one response. So these nodes, they are very important. Difference between the spatial and the temporal. Spatial summation is the summation of two nervous messages. These nervous messages, they came from different synapses. So spatial summation of two nervous messages, they come from different synapses. Whereas the temporal summation, temporal is the summation of two nervous messages, they come from only one synapse, from the same synapse. For the summation to take place, this message that are subjected to summation, they may be separated by short period of time. We can't give them, these messages should not be at the same time. If they are at the same time, we can summate them. So they should be separated by a short period of time in order to have the summation. So in this document, we described everything related to the temporal summation, the spatial summation, about the inhibitory synapses, excitatory synapses, EPSP, IPSP, and also we identified what happens when we have IPSP and what happens when we have EPSP. These notes are very important. So in this lecture, we solved part 1 in the probing the document, part 2, and in part 3, I skipped represent in a recording obtained in the case of maintenance summation of one of many fibers of type 1 and 2. This one I skip it, part A. And part B, name the type of summation obtained and justify your answer. Also, I skip it. I moved only to part C. Already these parts A and B, they are mentioned here. They are the same. So we don't want to waste time in drawing this. So part C, draw out the role of the motor neuron. It has a summation property. It receives many messages and add them to give one efferent message. This is the role of the motor neuron. So these nodes are very important to know them regarding the synapses.